Come on. My slides are not going. Okay, here we go. So my name is James Garner. I'm a PhD student at UMass Amherst, co-advised by Michelle Staudinger and Adrian Jordan. I have been fortunate to work with a number of different project partners and funding agencies and stakeholders, and including everyone and everything that you see here. A little bit about my background. Several years ago, I started my diadromous fish journey working as a biological fisheries technician with the Massachusetts Division of Marine Fisheries, where I fell in love with New England watersheds and diadromous fish. One of my favorite fish here, the sea lamprey, I happened to find one day when I had a piece of glass with me, so that was fun. And a couple of my colleagues working on a fyke net in the bottom left. The first year of my PhD, I worked part-time as the ecology program director for the Jones River Watershed Association, where these environmental DNA projects began. And I would like to take a moment to thank and acknowledge the Jones River Watershed Association for their dedication to ecological restoration, monitoring, and their collaborative spirit. Without my time and experience at the JRWA, none of these projects would be possible, and they will forever have my gratitude. Okay, so what is environmental DNA or eDNA? To put it simply, it's DNA sourced from environmental samples rather than directly from an organism itself. So let's say you were in this beautiful little stream. In that stream, there are dozens, if not hundreds, of different organisms within, several hundred yards. And all of those organisms are shedding DNA at different rates. If you were to take a sample of water from that, filter it, collect the DNA, you'd be able to tell things about different types of fish species, for example, plant communities, invertebrate communities, or even microbial communities within that, just from a single sample of water. I'm going to talk a little bit about how DNA gets into the water, and doing this work has fundamentally changed how I view swimming in any natural body of water. Essentially, everywhere you're just swimming in DNA soup. Organisms shed DNA at different rates and in different ways, whether it's sloughing off skin cells or scales, from excreting mucus or urine or things like that. And if you are anywhere near a herring run in the peak season, you're probably familiar with that picture in the bottom right of just herring scales everywhere. So how DNA gets in the water varies, and with different organisms have different rates of shedding depending on their physiology. So I'm going to walk you through a couple of my favorite examples of environmental DNA. The first of which is ancient DNA. And I think this is probably one of the coolest applications. You take a sediment core from a location with, for example, an ancient lake bed and extract DNA layer by layer. Using a technique like this, you can understand how biotic communities responded to past climate shifts or other detectable environmental stressors. And when I found this out, it just kind of blew my mind. And this could be applicable kind of anywhere there was ancient sediments that had no oxygen exposed to them. Water, I primarily work with water samples, and that could be anywhere from like an estuary to a fish ladder. I even have a colleague who samples bird baths in urban environments to see what kind of critters visit them. So it's very, very applicable in a lot of different ways. Air, I couldn't find a better picture for air sampling for eDNA. But this is essentially how it's done. And for example, a team in Copenhagen decided to test if environmental DNA could be extracted from the air. So they sampled outside of a local zoo using literally vacuum cleaners like you see and filters. And they were able to detect 25 of the 49 target species they were looking for. So they were able to detect lions in Copenhagen, which I thought was really interesting. And things like honey. Researchers were able to detect 9 bee pathogens and parasites just from sequencing honey samples for different beehives in a given region. And so really what this is saying is the limit to how you can use environmental DNA is your imagination. If there are organisms and you can sample some part of the environment, you could probably find environmental DNA. I like to think of it as a genetic nose for a given environment, just like a dog can detect minute particles of things that smell. Environmental DNA can detect minute traces of genetic material in a given environment. So it's important to think about what eDNA is actually telling us. And for a given time and a given place, eDNA can give us data about biodiversity, species presence, and relative abundance. And I like to think of, if you were taking one sample of eDNA, it's like taking a snapshot at that given time and place for whatever you might be looking for amongst these three things. And relative abundance gets that asterisk because it needs to be calibrated. And we'll talk a little bit more about that later. Okay, so I'm going to walk you through some potential applications and outcomes of eDNA monitoring. So the first application is detecting species presence. And the outcome is you can detect rare or endangered species or confirm that they are there. eDNA is incredibly good at finding trace amounts of genetic material and amplifying them and giving you a good signal. So finding those rare and endangered species is pretty a common application. Next, you can use eDNA to track non-indigenous species or invasive species. You can either use it to track current and ongoing invasions, or if you have a long-term data series, you might be able to detect a first encounter for a non-native species. And then from that, you can implement management protocols. If you have a long-term data set and in one place, you can start to detect shifts in phenology or species ranges in real time. And you can track species responses to human-induced stressors like climate change and habitat degradation. If you're doing biodiversity or community structure assessments, you can establish modern environmental DNA baselines. So you can get eDNA baselines that you can compare to traditional biodiversity monitoring techniques. Understanding ecosystem response to restoration or climate adaptation action. This is a picture of the Elm Street Dam removal in the Jones River watershed. Photo credit to Jimmy Powell, who's here today. And a lot of Jimmy's pictures are here. He does a great job. I love his photography. And you can use environmental DNA to track ecosystem responses to these restoration and adaptation actions in real time to see how community structure changes post restoration or dam removal or whatever it might be. Estimating species density or abundance. It provides a lower cost supplement or alternative to other methods. And again, these methods have to be calibrated, which we will touch on more later. Okay, incorporating environmental DNA into a work plan. The eDNA workflow has 4 basic steps. The first of which is sample collection. And this is a great point for communities to get involved, because any expert, citizen scientist, or volunteer can easily collect eDNA water samples. Again, perfect step for communities to get involved. And there's actually a really great protocol developed by the folks over at UMaine using Nestle Pure Life water bottles. They tested to see if Nestle Pure Life had any traces of DNA in them. They do not. And because of that, you can take just a store-bought water bottle, go to a sampling location, pour it out, and use these bottles as a sample collection device without ever having to sanitize or bleach any kind of Nalgene bottles, which you see me using here in this picture. Then there's the sample filtration. So you have DNA in the water, but you have to get that DNA onto something. And so you have to filter the water, excuse me, you have to filter the water and get it all in one place. And that's done through a variety of methods, but usually it's done with like a little filter disc where you collect, and once you have the DNA, you can then extract and wash the DNA to make sure that's the only thing that you have on that filter, and then you amplify or make copies of that DNA to then do your downstream laboratory analyses. The 4th step is analysis. So from the sample, you collect it, you filter it, you DNA extract it, And then you do your downstream analyses, either single species or multi-species analyses. And we'll talk a little bit more about that. We won't get into the nitty-gritty though. So this is a big figure, but the important part of this is that there are two main approaches to DNA, and this is kind of a DNA workflow. The main point of this is that there are, for both approaches, the first three steps are basically the same. The sample collection, the sample filtration and preservation, and the DNA extraction and amplification are the same. And from that point, you can decide what kind of downstream analyses you want to do. At the end of the DNA amplification stage, you're left with your DNA that you are going to analyze. And from there, you can do multiple approaches, the first of which is single species monitoring. After sample collection, filtration, and amplification, you can do something called quantitative PCR or qPCR. And this is simply using species-specific primers to capture target DNA. And a primer is just a short single-stranded DNA fragment that anneals or connects to target species-specific DNA you're looking for. So I like to imagine one half of a zipper, where you're looking for the other half of that zipper in your sample. And then this is what you can use to estimate species abundance. What you get at the end of this qPCR method is a concentration of DNA per unit volume. So you look within a stream, you filter, you extract, and it can tell you whether or not you have a trout in your stream, for example. Single species techniques are more sensitive to smaller genetic signals, and for capturing specific quantities of DNA, you're only searching for one genetic signature as opposed to looking for multiple in a large sample, which brings us to biodiversity or multi-species monitoring. And you do this using a technique known as eDNA metabarcoding. And it's used to assess biodiversity in a given location. And this uses universal primers that target all relevant species within a sample. And then once you have all of your DNA extracted and amplified and sequenced, you compare it to an online database with all known sequences. And from that, you can get matches and hits. And what that kind of looks like comes out like this. It looks like a large spreadsheet with all the species you got hits for. So in this table, where you see these common names, everywhere you see a common name is a fish that we got a positive hit for in some stretch of our watershed or somewhere. And this is only about a third of the species that we had. I just wanted to make it large enough for everybody to see. And so you get an understanding of community structure or biodiversity at a given time and place in your watershed. Okay, so what can eDNA tell us? It can tell us species presence, biodiversity, and estimates of abundance. A good way to think about eDNA is to consider exactly what your data is telling you compared to other traditional monitoring techniques. Consider a river. This is the Jones River watershed here, the headwaters here at Silver Lake, and it winds all the way down to Kingston Harbor. Again, consider a river. Downstream reaches are a collection of all upstream reaches. In order to parse out species source locations, multiple reaches and tributaries of the river have to be sampled. So if you wanted to know, what diadromous species, for example, this is a diadromous species diversity pre and post main stem dam removal chart, where in one part of your watershed, you'd have to sample upstream to be able to exclude your findings. And so just to kind of walk you through this figure, each location there are 19 locations. In the blue, it's the post-restoration monitoring diadromous fish diversity, and in the yellow, it's our kind of control. And this is monitoring after the Elm Street Dam removal, which is where this red line is roughly, took place in 2019. It was a main stem head of tide dam that was taken out by the Jones River Watershed Association. And the yellow pie sections are taken in February of 2020, so directly after the Elm Street Dam removal. And in all locations except for two, obviously, the diadromous fish biodiversity increased even well upstream of the Elm Street Dam. An interesting thing to notice or to note And something that I found supremely interesting is the two locations where the biodiversity did not increase within the Jones River watershed had known, or at least one smelt brook here had a known culvert that was hard for diadermous species to bypass. And then here at Jones River Brook, the Massachusetts Division of Marine Fisheries found a log jam of sorts downstream, which made passage very hard. So it can also help you identify locations where you might need to do extra restoration actions, or where impediments to passage might be. Fun fact. Okay, so consider what your data is telling you compared to other traditional monitoring techniques, and what are some other ways that we monitor aquatic life. Volunteer herring counts gives us an estimation of river herring run size for a season or abundance for a given watershed. Electrofishing surveys, they can tell us about the biodiversity of a given stretch, or you can use that by measuring catch per unit effort to get at abundance or density. And something like a FICENet survey, which can help you document biological characteristics of species like rainbow smelt. It can also give you estimates for abundance. And the nice thing about techniques like FICENet surveys and electrofishing surveys is it allows you to collect population data, population-specific data like meristics. You get to measure the fish that you're catching, which brings us to things that eDNA can't tell you. It can't tell you absolute abundance. It can't tell you the age and growth structure for fish populations. And it can't tell you what life stage your DNA signal came from, at least not yet. And for context, you don't know if your signal's coming from gametes. You don't know if it's coming from a juvenile fish or an adult fish or a dead fish rotting upstream. So it does have some biases. And the way I like to think about it is that eDNA is a supplement to ongoing monitoring practices, not a replacement. So let's go over some strengths and weaknesses of eDNA techniques. The pros: it's orders of magnitude more cost effective than implementing an alternative monitoring strategy most of the time. It's less upfront effort. To begin doing these surveys, all you have to do is dip a bottle of water and freeze it. And that might be just throwing it in a chest freezer until you have the funds to do the downstream sampling or send samples off. It's more accessible than traditional ecological monitoring. My favorite part about eDNA is that it's non-invasive and non-destructive. You don't have to catch, harass, or kill any of the species that you're looking to learn about. Rare, shy, or cryptic or well-hidden species typically show up pretty well using environmental DNA versus established techniques, especially if you aren't willing to spend hours and hours and hours searching for them. The field is constantly being refined. Every week I read a really new, really cool paper that I'm just, stunned about, and novel applications are emerging regularly. The cons: false positives and negatives are common. My favorite anecdote is I got a hit for a bluefin tuna 3/4 upstream in the Jones River watershed, and I know for a fact there are no bluefin tuna there. And my best guess is somebody had some tuna for lunch, ate it, and it came out through their septic system, which kind of speaks to the power of eDNA because it went through multiple filters. It went through the human filter, it went through the septic filter, and this technique is still powerful enough to detect that signal after all of that. It can be controversial, especially when it comes to abundance metrics. They always have to be calibrated, and it has to be done carefully. The barrier to entry to downstream analyses can be extremely high. So the first two steps, the collection and filtration, are pretty simple to do, but the downstream extractions and analysis usually requires sending samples off to a lab or having a lot of high-tech equipment. And the field still needs a fair amount of refinement. So known biases. eDNA monitoring techniques have limits and sampling biases. As you can see here, this is a hagfish. It's a jawless fish that when caught exudes a lot of, I don't have a better word to say than slime. And a hagfish is going to have a much larger DNA signal than, for example, a green crab. And so those rates of DNA shedding have to be accounted for in your sampling design and your analyses. Environmental biases, every aquatic system is different, and those differences have to be accounted for. The reason this image is here is that every step along the way, you introduce an opportunity for contamination. And contamination is a big problem in DNA, in environmental DNA. You would not want to store your sample or process a sample in a room that has any of the species that you know you're looking for. So you have to be pretty careful. The importance of calibration. So eDNA samples of both varieties give you what's known as a read count score, or a concentration of DNA per unit volume of water. Single species methods are more sensitive and give you a higher degree of resolution compared to multi-species approaches, because they're only looking for and amplifying the DNA from one species, as opposed to amplifying everything in a sample and processing all of it. In the graph to the right, we see a concentration of eDNA signal on the y-axis and the number of carp per meter squared on the x-axis, calculated from a known number of carp within a concrete pond. Excuse me. As you can see, we have a positive correlation between eDNA concentration and number of carp per meter squared. It's not perfect, however, it's pretty good. And these methods are constantly being refined and updated. One more time, just to drive it home. eDNA monitoring techniques are not meant to replace other monitoring strategies, but to enhance ongoing monitoring efforts, or provide an accessible and affordable starting point for watershed, biodiversity, and species monitoring efforts. I like to think of eDNA as a force multiplier for other monitoring efforts. eDNA can help you fill in knowledge gaps where you might not be able to answer certain questions using traditional methods. Okay, so you know what eDNA is, you know kind of how to do it. Let's go over some potential applications for river herring management and restoration. One of my favorite applications is determining if your restoration or adaptation action has met its intended goals, especially if its intended goal is to increase biodiversity upstream. And it doesn't matter what the restoration action or adaptation action is. If the goal is to increase biodiversity, eDNA would be a great way to compare pre and post restoration monitoring. Determining how far upstream river herring are making it into your watershed. If you just removed a dam, like where the Elm Street Dam was removed, and you want to know how far fish are penetrating or where fish are getting to in your watershed, all it takes is going out and collecting a sample of water, freezing it, and sending it off, and you can get an answer relatively quickly. Whether or not river herring are making it upstream of fish ladders. One of my projects, in collaboration with the Massachusetts Division of Marine Fisheries and the North and South River Watershed Associations, is calibrating environmental DNA methods to electrofishing surveys for American shad. And this survey starts a couple hundred yards downstream of a dam, and we work our way up. And the second eDNA sample that we take is just upstream of the dam, so that we can answer the question for our stakeholders if shad are making it upstream of those dams currently. And once we get results, which hopefully will come soon, we'll be able to answer that for them. If you have a long-term data set, you can answer when exactly river herring are arriving to your watershed, how long they're staying, and when they've all left if you sample through time. And so if you have a long enough data set, you could start answering questions about phenology. You could start looking at when alewife get there versus blueback or American eel or whoever you're looking for. And the longer that you have this going, the more questions you can really answer. And so one of my projects is with the town of Plymouth, and we're going to start year-round sampling every 2 weeks. And so we'll be able to kind of get at some of these questions within a year or two. If river herring are entering your watershed from other locations. So this is Silver Lake, the headwaters of the Jones River. The Jones River comes out right here. And something I found interesting when I was doing this was that I got signals for river herring coming in from Tubbs Brook, which flows into Silver Lake. And so you can identify where fish are getting into your watershed, depending on what's going on in your watershed. One of my favorites is biodiversity and community structure assessments. You can get a feel for what your prey species in a given location look like for river herring, or for whatever species you're interested in monitoring, what your plant community structures look like, or your habitat looks like. You can get a feel for aquatic pathogens or freshwater competitors. If you know you have invasive species in your habitat, you can get a feel for how those species communities are changing over time. Are their populations growing? If you're doing multiple sampling efforts through time, and if you have a long-term data set, you might be able to detect the first time an invasive species gets to your watershed, which hopefully doesn't happen, but it's a possible application. And something that I'm really hoping to figure out more about is to provide low-cost biological evidence for permitting. I'm currently in talks with different permitting agencies and seeing if environmental DNA would be a viable solution to provide biological evidence for specific permits. Things to consider: what monitoring is already going on in your watershed? What questions are you asking, and where are your knowledge gaps? What are the hydrologic characteristics of your watershed? Is it a lake? Is it a pond? Is it a stream, estuary, or marine? Each one of those types of habitats is going to require a different sampling strategy, which is incredibly important to consider if you want to effectively answer your questions. Are you looking for one species or many? And are you taking a snapshot or a single sample at a single place and time? Or are you collecting a time series of data? I like to think of eDNA like point sampling is a snapshot. And if you're taking multiple snapshots, it eventually builds up to be a motion picture of a given ecosystem. All right, so you've collected your water samples, you filtered them. Now what? You can build your own capacity. You can invest in a qPCR or single species testing kit. There are many field or turnkey kits available. These kits can be a little bit complicated to implement, but they're not impossible, and they're incredibly effective. They can be also quite expensive, as opposed to just collecting samples and sending them off. If you're interested in this approach, a Google search for eDNA field sampler will yield several very good options. Alternatively, you can ship frozen water samples or preserved filtered samples to a lab. And when you do that, usually those laboratories will process everything and kind of give you a write-up and tell you what you're looking at. Some New England-based labs that you can work with. I've had great success working with the University of Maine Kinnison Lab. I have colleagues who have worked with UNH, the Hubbard Center for Genome Studies, and there are a handful of other really great environmental DNA labs that you could work with. And if you want this information, please feel free to contact me via email after this talk. I'd be happy to send you any information from this talk. Okay, so what's it going to cost me? Biodiversity or metabarcoding costs roughly $2,000, between $2,000 and $3,000 for 96 samples. And why 96 samples? The equipment that you use to do metabarcoding is limited to 96 samples on the machine that you use to sequence all the DNA. And that machine costs about $2,000 to $3,000 to run those 96 samples, which is why it's limited to 96 and costs that much. For qPCR or single species approaches, it's less than $20 per sample, and that price drops the more samples that you include. Okay, so some of my ongoing work. To the left is a map of New England, and everywhere you see a colored dot is somewhere that either I or a collaborator has a project going on. The pink dots or the salmon dots are where the Jones River watershed is, and the yellow, gray, and blue dots are where eDNA calibration studies are going on. The goal of my PhD research is to make eDNA tools and techniques more accessible and to get them in the hands of the people, communities, and managers who can use them the most. So my first project is using environmental DNA biodiversity methods to measure restoration action efficacy in coastal New England watersheds. That's the Jones River watershed. After the Elm Street Dam removal in 2019, we started this eDNA survey in 2020, and we've continued every year, or I have continued every year and I will continue doing this until we have reached a point of understandable equilibrium within the Jones River watershed. And so this is a super exciting project, and once I find a little bit more funding for it, we'll get results for 2021 and 2022. Project 2, calibrating eDNA abundance metrics to establish methods. So electronic hair encounters, video hair encounters, volunteer hair encounters, FICENet surveys, juvenile purse seine surveys, and electrofishing surveys all throughout the region. Detecting population-specific, river-specific, genetic signatures of American shad using environmental DNA techniques that are currently being developed by my partners at the DMF and a couple other collaborators. This is super exciting because being able to detect different populations of fish within the same species that might need different managing protocols is super exciting, at least to me. And project 4, developing a field deployable, affordable, equitable, and accessible eDNA biodiversity monitoring kit. This project is being tested and validated in conjunction with the town of Plymouth, Massachusetts, in preparation for an upcoming dam bypass project. And the great thing about Plymouth is we are going to be doing year-round monitoring in that watershed. So eventually we'll be able to answer some really cool questions about how fish are interacting with the town brook system. So here's a short summary of my 4 projects. Here are some resources and literature I'd be happy to send you if you have any questions. And with that, I will take any questions. And again, contact information, jggarner@umass.edu, and thank you for listening to me.
Thank you so much, James. I love your analogies. This is really complicated stuff, and it's really helpful to have those little reminders, especially the primer is the other half of the zipper. Perfect. So thank you so much for the overview. If folks have questions, you can type them into the chat box, and then I can read them off to James.
Should I stop sharing or what?
You know what, let's leave it on for a second, just in case somebody wants you to go back to a previous slide. So first question, after you filter the sample, what percentage of the filtered stuff is actually DNA?
That's a great question. It depends on the river and it depends on the system or the lake or what have you. If you have a really high sediment load system, I would say like the majority of it's going to be sediment. But the nice thing about that is that DNA attaches to small particles in the water. So if you have a lot of sediment in your water column, that sediment's going to collect DNA for you. So that's another environmental variable that kind of has to be considered when you're doing this. If you have really clean water, you're still going to be getting a lot of DNA, but it might not be the same amount as another system. And like I mentioned before, every system's unique, and you have to consider different hydrologic or geologic factors in order to get an accurate representation of what's going on.
Thank you. Next one, can this be used to certify vernal pools? Not herring related, but important watershed question.
Yeah, if you're looking for like a salamander species in a vernal pool, yeah, absolutely. That's a great application.
Yeah, and I think some of the first papers on eDNA I ever read were about trying to find really rare salamander species in Appalachian streams, but they didn't want to go disturb the salamanders so they could find their eDNA in the water.
Yeah, every time I go hiking, I want to sample every vernal pool I see, and I have to stop myself because I don't have that much money.
How fast can turnaround time be for samples at the lab? So could eDNA be used for real-time detection of the arrival of a species?
Another great question, and it depends. If you are in a full-on genomics lab, you could do it same day, real time. If you buy one of those turnkey kits and you're only using a single species primer, you can also get same day turnaround. However, if you don't have access to a genomics lab and you're sending samples out, and it depends because COVID really messed up scientific supply chains. So it could be anywhere from, with the supply chain up, a turnaround of a couple of weeks. But right now, I think that deadline has been shifted a little bit, but I'm hoping, knock on wood, that will come down pretty soon. So if you have resources or the equipment, quickly, if you have to ship them out, I would say within 2 weeks to a couple of months.
All right, thank you. Next question is, do you have let's see, so many questions coming in. Question disappeared. Okay, do you have a rough idea of how many meters of river would be covered in one sample with reasonable accuracy? So like 50 meters, 1,000 meters or more?
That's a great question. And it really depends on the depth of the river, the flow rate of the river. For reference, there have been studies done where they've taken like a caged experiment with an organism that wasn't in that system at all, and detected how good their signal strength was all the way downstream. And you can get hits miles downstream. However, a really interesting thing about a river is that you have DNA kind of follows in a plume. So it's not going to, in a flowing system, it's not going to spread out throughout that entire system. It's going to follow kind of a straight line trajectory downstream most of the time, depending on the hydrologics. So it's it depends, I guess, is the answer. So sometimes many miles, sometimes that signal strength will disperse after a couple hundred yards. The traditional thinking is your first detection of a species is probably going to be within 50 yards of the sample you're targeting, and then up to several miles downstream. So if you wanted to rule out different tributaries, you could take one sample downstream of two tributaries, one sample upstream, and see what differences you get there, to see if what's being included from different tributaries, something like that. I hope that made sense.
All right, thank you. Next one is, can eDNA differentiate between alewife and blueback, the two river herring species?
That's a great question, and I think that is currently being worked on right now in a metabarcoding sense. You do get both alewife and blueback signatures. I don't know how accurate those are since they're so closely related. I don't think a species-specific primer has been developed that gives you really good results yet, but I do know that there are groups working on that right now.
John Shepard with Division of Marine Fisheries just typed in that for differentiating alewife versus blueback, Roger Roelofsen, who's a researcher at Eastern Carolina University, has conducted a study to detect both species, but he's not sure if he's published the work yet. So we'll look for the papers. If there's any, we can send them out on the list.
Short and long of it, soon, hopefully.
Yeah. How long can frozen samples be stored, and does the DNA degrade over time?
That's a great question. So DNA is a pretty stable molecule. However, things like temperature, UV light, enzyme action, things like that all contribute to that DNA degrading. And so if you get a frozen sample in a freezer, it's relatively stable. You can keep it for, up to a year or two before, I don't think anyone's ever held on to enough water to see how long DNA signals persist, but the way that people go about it is saying, the sooner you filter it, the better. So it's safer to do it more quickly. However, I do think that you will get a good signal even if you freeze it for up to a year or more. But yeah, it's wiser to do it more quickly.
All right, thank you. We'll get a couple more. Ben Gehagen chimed in on the vernal pool question and said also that, let's see, right now, Natural Heritage or Department of Environmental Protection will not confirm the existence of a vernal pool just from species presence, that, there's a bunch of other things, but as well. So it's one piece of the puzzle. Do you think eDNA could be used to distinguish between a European eel and an American eel?
I do not know the answer to that question.
Interesting.
I would have to see what primers exist and how different they are. I mean, that's, one of the main applications is species detection. So I would imagine that it's possible. I do not know the state of it right now.
So that was a lot of questions. Thank you very much, James. I think we'll stop Q&A for James now, and we will transition over to Eric's presentation. So thank you so much, James. That was great. And hopefully you'll start to get some questions from folks about how to implement this in their watersheds.
Thanks,
Okay.
everybody.
So, and if you've got more questions for James, you can just keep typing them in the box as you come up with them. And then we can come back to James at the end. So